Hello, today we are going to talk about genetic sequencing. And first of all, it's important to understand that the difference between a common biochemistry analysis test, for example, glucose testing or cholesterol testing, where we always find a number showing that the value is above normal or below normal for glucose or cholesterol or any other test that we usually do. So in genetic testing, what we find in sequencing is a sequence of letters that represent the nucleotides that form our DNA. So we have 4 letters, A, C, T, and G, and we can have any sequence of those letters representing our DNA, and we can sequence just one gene, or we can sequence all of our genome. What we find is this whole amount of letters in a certain sequence, and what we have to do to give a result, to have a result, to understand, to interpret what we have in our hands, we have to compare to a database. A database of what? A database of many other patients that don't have any diseases. And then we have to point variations in our patient's DNA, and then we have to try to understand if the variations that we found are variations that may be disease-causing or not, or just what we call polymorphisms, which would be variations from normal but not disease-causing. And to help us, we have bioinformatic tools that can try to predict and give us a probability number if that variation may be disease-causing or not. So this is a limitation of the test. Once we do a broad genetic testing, we not always have in our hands a result that we can call positive or negative. We have to interpret it. And we may have to take other steps in order to help us with the interpretation. For example, we may have to do linkage analysis and sequence our patients' parents or relatives in order to compare the variations that we found in our patient with variations that may be present in relatives or parents of the patient that are not affected, and also compare with parents or relatives that may be affected as well. So that may help us with what we call linkage analysis. Also very important to understand when we analyze a genetic testing, a sequencing, is our database. It's not as simple as, oh, I have a database and I'm going to sequence this patient. No, you have to understand that ethnicity is very important. So variations among certain specific populations may be more common in that population and not as common in another ethnical population. So when we have a patient and we know the patient's ethnicity, it's much better and it will give us a more accurate result if we can find a database with the same ethnic background as our patient. So that is also very important to understand. And also another limitation of genetic testing that we have to keep in mind is that not all the genetic mutations are present in the coding sequence of our DNA that we call exons. Some mutations, some disease-causing variations are found in what we call introns, which are parts of our DNA that are not coding regions for proteins. And we have also to keep in mind that the introns are not the deep introns are not sequenced on a regular basis in a commercial laboratory. We may sequence introns, but for that we have to plan ahead and make it in a research facility, in a research setting, and also we have to create a whole new database just for the intron sequencing. And once we usually when we do that, we select just a gene or two or three, not a high number of genes to sequence introns, because it represents almost 90 times more sequencing than sequencing just our coding regions. So those are the limitations and how we have to interpret genetic sequencing.